go. We've gone for polar and nonpolar molecules. Now let's look at something else that's not too much emphasized in your typical chemistry class, but it has a great deal of importance in biology. Biology depends on these particular types of, they're called a type of bond, but they're really not the type of chemical bonds we had before. They're actually attractions between opposite charges. But they're not like an ionic and covalent bond where they're actually trying to fill their electron shells. But these bonds, and they are also extremely weak. These bonds, called hydrogen bonds, despite being very weak, are very, very important. Now let's make a hydrogen bond. We're going to use water as an example because water is very good at making hydrogen bonds with other water molecules. And then we'll generalize that because not only are hydrogen bonds important in the properties of water, they're also important in many other aspects of biology. So let's take a look at this. Let's take a look at a water molecule. Now water molecules, of course, are very polar molecules. So we have that partial negative charges here, partial positive charges here. Now let's suppose another water molecule comes tumbling on by. Let's suppose we have the oxygen in that water over here. These partial charges are opposite, so they will attract each other. It's not a very strong attraction because those are only partial rather than full charges, but they do attract each other. I'm going to represent a dotted line to represent that attraction. That's what we have called a hydrogen bond. It's an attraction between this positive charge on the hydrogen and this negative charge on the oxygen, at least in this case. Now, once again, we could have another oxygen, another water molecule here, and we could get a weak hydrogen bond here. And if we had an oxygen over here, a water here, we could get weak hydrogen bonds here, and so on. And we can continue building this up to make a network of hydrogen bonds. Now, in the case of water, that's what keeps water liquid. Because at room temperature, in liquid water, those molecules are moving fast enough that a hydrogen bond will form from one water molecule to the next, but will be only temporary because after a while, that other water molecule is going to be moving away fast enough so the hydrogen bond breaks. But it might make a hydrogen bond with another nearby water molecule. So when we look at liquid water, the hydrogen bonds, it's a constantly changing network of hydrogen bonds. As one water molecule passes the other, forms a hydrogen bond temporarily, and then breaks as it moves on. And that's what liquid water is. Now, if you cool water down, the motion of the molecules starts to slow down. So what happens in this case, eventually the water molecules are moving so slowly that when they form a hydrogen bond with another water molecule, they're not moving fast enough to break it. So they stay in place. And then more water molecules come in and also form hydrogen bonds, and they stay in place. And pretty soon that liquid water turns into solid water. Ice. Ice is a network of water molecules, and the hydrogen bonds are holding it together in the form of a solid. Now, something unique about water, because the shape of the water molecule is kind of shaped like a triangle. When water turns into ice, it actually takes up more volume, about 10% more volume than water as a liquid, where the molecules are tumbling around in every which way. So ice is one of the few substances that expands when it freezes. Almost every other thing we know of, the liquid actually, when something freezes, it actually has a higher density than the liquid state. So like if I take steel or something and melt it down, I add molten steel and I throw a piece of steel into it, a piece of iron into that, it will sink to the bottom before it melts away. But water, when it freezes, it actually floats. And that's because of the shape of the water molecule. But what we call ice is actually water held together by hydrogen bonds. No longer are they breaking. Okay, so water forms hydrogen bonds with each other. Now we could also look at ammonia. Ammonia is a similar kind of thing. We have these partial positive charges on each of the hydrogens, and they can form hydrogen bonds with other ammonias. And I'm just making one here. So ammonia forms hydrogen bonds with other ammonia molecules. And ammonia molecules can form hydrogen bonds with water molecules as well. Now an interesting thing here, because oxygen is much more electronegative than nitrogen is, the partial charges here are more intense than they are in the case of ammonia. So that means the hydrogen bonds that water makes with each other are stronger than the hydrogen bonds that ammonia makes with each other. Keep that point in mind here. Now hydrogen bonds are overall quite weak. They're the weakest things that we see. Matter of fact, the bond strengths are usually a few kilocalories per mole. What that means in real terms is that most things that are held together by hydrogen bonds, if you heat them up to, the boiling point of water, or in many cases quite a bit less, the hydrogen bonds break. So it doesn't take, in fact, that's what happens when you boil water. You heat water up, and then the hydrogen bonds break, and some of those water molecules pop off into the air as water vapor. All right. And many hydrogen bonds are weaker than that. Water makes fairly strong hydrogen bonds with each other. So in most cases, hydrogen bonds are weaker than that. In many cases, temperatures that are known, especially biologically important molecules, temperatures that are no more than a hot summer day down here, can break hydrogen bonds in critical molecules and cause all kinds of biological problems. Okay, so we have these weak hydrogen bonds. And it holds water together, and ammonia together, things like that. Now, it turns out, of course, if you look at water, water has, and this is mostly because of the hydrogen bond formation, the fairly tight hydrogen bonds that water makes. Water's boiling point is 100 Celsius, freezing point is 0. Now, what about ammonia? Ammonia doesn't make hydrogen bonds. It makes hydrogen bonds, but not as strong as the hydrogen bonds between water molecules. So what would you expect the boiling point of ammonia to be? Less or more than that of water? Less, considerably less. Ammonia is a gas at room temperature and ordinary pressure. And ordinary atmospheric pressure to liquefy ammonia, you have to go down to about minus 50 or 60 degrees Celsius. In other words, about what you'd find in a typical Antarctic winter, because it makes weaker hydrogen bonds. Now, one thing is ammonia and water have a very similar molecular weight. 18 here, 17 here. Now, to continue this on, let's take a molecule with a molecular weight of 16 that does not form hydrogen bonds with each other. The reason why is there's no partial charges on these hydrogens or on the carbons. They're sharing electrons evenly. So this is methane. Now at atmospheric pressure, how cold do you have to get to liquefy methane? Well, if you want liquid methane, in fact whole lakes of it, go and take a spaceship and fly out to Saturn. And that moon of Saturn called Titan has lakes of liquid methane at a nice balmy 190 below 0 Celsius. And it's got an atmosphere a little bit higher pressure than ours. But to liquefy methane, you have to go down to cryogenic temperatures. And even though it's about the same size, same molecular weight as the others, methane does not form hydrogen bonds with other methane molecules. There's no charges. So methane has a very low boiling point. Ammonia, which forms somewhat weak hydrogen bonds with each other, has a somewhat higher boiling point. And water has, for a small molecule, a very high boiling point. And that's actually important, as we'll see later on. Okay, so one of the reasons that we have liquid water at this kind of temperature and not at cryogenic temperatures is the hydrogen bonds hold water together to keep it liquid at these kinds of temperatures. Okay, so clearly hydrogen bonds play important roles in things like water and stuff like that, but we also find hydrogen bonds playing roles in other types of things as well. And we'll see that, but first let's kind of generalize. How do we make a hydrogen bond? Well, when you, excuse me? Oh, okay. You gotta have a couple different ingredients to make a hydrogen bond. First of all, you have to have a hydrogen. Okay? You have to have a hydrogen atom. It wouldn't be called a hydrogen bond if hydrogen had nothing to do with it, right? But it can't just be any hydrogen atom. It has to be a hydrogen atom that has a partial positive electrical charge on it. Now recall we looked at water and ammonia, and those hydrogens on those molecules had partial positive charges. On the other hand, methane, which does not form hydrogen bonds, it has hydrogens on it, but they have no charge. So you need a partial positive charge on a hydrogen atom. So hydrogen with partial positive charge. Now, how do you get a partial positive charge on hydrogens? You have to covalently bond it to something.
Like fluorine.
Excuse me?
Like fluorine.
Like fluorine, like oxygen, like nitrogen, but not like carbon. What's the difference between those folks?
Electronegativity.
Excuse me?
Electronegativity.
Electronegative. You have to covalently bond to something that's a lot more electronegative than you are. So, we'll say covalently bonded to electronegative atom. And examples include things like fluorine, chlorine, oxygen, nitrogen, and a few other things. Okay, so that's ingredient one to make hydrogen bond. Hydrogen with a partial positive charge because it's attached to something a lot more electronegative than it is. Now the second thing you need is you need something with a partial negative charge. And that's going to be an electronegative atom like oxygen or to a lesser extent nitrogen. And it has to be attached to something that's a lot less electronegative than it is, like carbon or hydrogen or what have you. So that way, it takes an uneven share of those shared pairs of electrons, and it gets a partial negative charge. So ingredient number two, electronegative atom. And it's covalently bonded to something a lot less electronegative. Okay, that's ingredient number two. Now, third thing, ingredient number three to make hydrogen bonds is you have to be physically close to each other. You can have hydrogen bond forming groups, two groups that could potentially form hydrogen bonds, but because the attraction's weak to begin with, they have to, and attraction falls off with the square of the distance. You're twice as far away, you got a quarter of the strength of those hydrogen bonds. So pretty soon it shrinks down to virtually nothing. You have to get the hydrogen bond forming groups within about an atomic diameter of each other. They have to be close together. If they're, several atomic diameters apart, it's not going to happen. The attraction is simply too weak. So they've got to be physically close together, close enough for the hydrogen bond to actually form. Okay. So those are the three ingredients we need to make hydrogen bonds. Now a little exercise that we'll do here in class. I'm going to put a couple different parts of molecules, chemical or functional groups and stuff, and we're going to assume that we put them close enough together. Question is, Will hydrogen bonds form or will they not? And why or why not? So let's do that. I'm going to put right over here a carbon with a double bond to an oxygen. And then over here a nitrogen and a hydrogen and a nitrogen and going off to something else. Would we get hydrogen bonds forming between these guys? Yes or no?
Yes.
Okay, we have to look and see for the key ingredients. First, let's look if we satisfy condition 1. Hydrogen with a partial positive charge because it's attached to something a lot more electronegative. Hydrogen and nitrogen, yes, it's going to have a partial positive charge here. Ingredient number 2, an electronegative atom that has a partial negative charge because it's covalently bonded to something a lot less than that electronegative. Oxygen making a double covalent bond with carbon. Oxygen far more electronegative than carbon. So absolutely we'll have a partial negative charge over here and a hydrogen bond does indeed form. Okay. Now let's see what. Okay, when we get hydrogen bonds forming here, let's look and see if we've satisfied the conditions. First, let's look at this hydrogen. Does it have a partial positive charge? Yes, because it's attached to nitrogen. So I have a partial positive charge here. What about this guy? Does this have a partial negative charge? Well, it's attached with a triple covalent bond to carbon, and it is more electronegative, a fair amount more than carbon, so it will have a partial negative charge. And yes, we will get hydrogen bonds here. It'll be weaker, it'll be somewhat weaker than the one we see up here, but it'll be there nevertheless. Now, what about this one? Will we get hydrogen bonds here? People shaking their head saying, no, no, no. In this case, the nays do indeed have it. Because over here, we got hydrogen, but it's attached to carbon. They're equally electronegative, so this guy has no charge on it. Meanwhile, we do have a partial negative charge on that oxygen because it's covalently bonded to carbon. But it's got nothing to be attracted to. It's all dressed up and nowhere to go in this case. So hydrogen bonds will not form in that example. Now, by the way, I often have a test question on it. Which group or groups can form hydrogen bonds? I often do that on a test. No guarantees, but common question, common fight. Okay, let's see. What about here? All right. Hydrogen bonds forming here?
No, there's not.
Absolutely not. First of all, there's no hydrogen at all. Okay. Second of all, let's look at the charges. Oxygen has a partial negative charge, and so does nitrogen. They're actually not only won't they attract each other, they will repel each other because the charges are the same. So you get those two groups together, you're going to push each other away. Okay, so those are some examples of hydrogen bonds and how to make them. Now, in many of the organic molecules that are components of things like nucleic acids and proteins, there are hydrogen bond forming groups in there. And many times hydrogen bonds will indeed form if they're close enough together. And that makes hydrogen bonds especially important for life. Despite their weakness, they play critical biochemical roles. So let's take a little look on this. Is it okay if I erase your little ingredient list to make a hydrogen bond? All right. So where do we find hydrogen bonds? One of the most important things is nucleic acids. In other words, DNA and RNA. Because it turns out nucleic acid, you have a phosphate ion connected to a 5-carbon sugar, and then coming off that sugar is something called a base. It's called a base because it makes an alkaline or basic solution if you throw it in water. Bases are polar molecules that are rich in nitrogen. They're rings, single or double rings, and they're rich in nitrogen. They have all kinds. They have nitrogens connected to hydrogens, oxygens connected to hydrogens, and things like that. So you have a lot of various kinds of polar groups connected to these bases. And there's, at least in nucleic acids, 4 major kinds of bases, or 5 actually. Something called adenine, or A, something called cytosine, or C, something called guanine, or G, something called thymine, which is found in DNA only, or T, and a very similar molecule called uracil, which is found in RNA. Now these 5 bases, they all have things that can potentially form hydrogen bonds with each other. The problem is the shapes of the molecules allow only certain combinations between certain bases where they can actually fit close enough together where hydrogen bonds will actually form. Other combinations, the potential groups are just too far apart. And the combinations that can form hydrogen bonds with each other are going to be adenine can form 2 hydrogen bonds with thymine or 2 hydrogen bonds with uracil. Both of those, thymine and uracil, are very, very similar molecules. And cytosine can form 3 hydrogen bonds with guanine. So adenine and thymine hold together a little bit less tightly than cytosine and guanine. Okay, now, if you took something like cytosine and adenine, there are potential hydrogen bond forming groups, but the molecules can't fit to get those groups close enough together. So hydrogen bonds will not form between those bases. These are the only combinations of bases that can form hydrogen bonds with each other. Now, why is that important? Well, if you take a look at a DNA molecule, a DNA molecule is a double strand, so wound around each other in a spiral. They call it the double helix. Helix is just a spiral coil. And you have the structure of sugars and phosphates, I'm drawing as a line, and then coming off you have these different bases. Like so. Now, the second strand of DNA sticks together because it has a sequence of bases that can form hydrogen bonds with the first one. So we get hydrogen bonds forming here, 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 and so on. And that holds the strands of DNA together. Now, because hydrogen bonds are weak, if you heat that DNA up to temperatures anywhere from about 70 to about 95 degrees Celsius, the heat will break the hydrogen bonds and the strands will separate apart. In fact, that's widely used in a technique called PCR, a common molecular biology technique, the polymerase chain reaction. You separate the strands of DNA each cycle by heating them up close to the boiling point and then cooling them down, and then enzymes copy each strand of DNA. Okay, and that's the critical thing about it. The hydrogen bonds hold the DNA strand together, but let's physically separate these strands. Let's pull these strands apart, whether it's by heat or in a living cell, enzymes will do that. So we have T over here, A, C, G, T, and A right over here. Okay, we pull the 2 strands apart. Now, enzymes can take what we call nucleotides. These are subunits that include a base. They have a sugar, a phosphate, and a base. And we can use the ability to form hydrogen bonds to dot, quote, the appropriate base. This representing individual nucleotides. And then we glue them all together, and we do the same thing down here. And we glue all these guys together. And what have we just done? We have replicated DNA. We have two identical strands here. We start out with one. We pull the strands apart. One strand serves as a guide, or what they call template, those of you who remember genetics, to get accurate synthesis of the other strand. And that's one of the most fundamental properties of life. Hydrogen bonds ensure the accuracy of that process. It would do you no good if you had a nice set of DNA, and then when the cell divides, you make all kinds of mistakes and mutations. Hydrogen bonds give an accuracy rate of about 1 mistake in 1,000,000. And then there's enzymes that are called proofreading enzymes, kind of like the spell checker and a word processor. The occasional mistake, and I'll just do a little example here. Let's suppose we screwed up here and we stuck a thymine over here. Well, thymine would not be able to form a hydrogen bond, and what would happen is you actually get a little loop out of a piece of DNA. The proofreading enzyme recognizes that, oh boy, somebody screwed up, and then it goes and gets rid of that offending base and sticks the right one in. And that gives you another 1,000-fold increase in accuracy. Put it this way, our human genome, or genetic blueprint, has about 3.2 billion pairs of bases. When a cell divides, typically you only have a couple mistakes every time the cell divides. That would be like copying several hundred copies of our textbook and making only 1 or 2 single-letter mistakes in the whole operation. That's pretty darn impressive. But hydrogen bonds are critical for that process. Without hydrogen bonds, you simply would not get that level of accuracy. Dividing cells would quickly generate all kinds of mutations, and eventually the daughter cells would die, the descendants would die. And that would be it for life on this planet. Okay, so DNA replication is critically dependent on hydrogen bonds, but that's not the only thing. Let's suppose we got rid of a replication, and we pull the two strands, our original strands of DNA apart, and now enzymes add nucleotides that make up the RNA molecule. So we have T over here, we're going to have nucleotide with A. We're going to have U over here, uracil. Uracil is found in RNA, it's very similar to thymine, and makes hydrogen bonds here. And then we have guanine, cytosine, adenine, and uracil again. Now, of course, DNA, that's the master storehouse of genetic information, but what actually does all the functioning of DNA in the cell are these different kinds of RNA molecules, the messenger RNAs, the transfer RNAs, and so on. All RNAs are synthesized using a gene, a stretch of DNA that contains the instructions to make that RNA. So you could have a perfectly good set of DNA, and if you made mistakes in RNA synthesis, you'd still be up the creek. But once again, hydrogen bonds come to the rescue. They ensure a 1 mistake in 1,000,000 level of accuracy. So that's actually very good. So we see in life, hydrogen bonds play a critical role in genetics, in accurate DNA replication, and in accurate RNA synthesis. Okay, that's 2 things. But there's still more. The other thing that hydrogen bonds do hydrogen bonds contribute to the structure of proteins. Now, as we're going to see when we talk about proteins, the function of proteins and the regulation of proteins are critically dependent on the exact 3-dimensional structure of those proteins. If you alter the structure of a protein, you're going to alter its function. You may kill its function off entirely. One of the contributing factors to that critically important protein structure are hydrogen bonds. That's not the only factor. In fact, it's not even the major factor, but it does contribute. So we see hydrogen bonds playing roles in protein structure and in accurate DNA replication and RNA synthesis. Those are some of the most fundamental properties of life on this planet. They're absolutely essential. So hydrogen bonds, we would not be here without hydrogen bonds. Even though they're very weak and they don't play too much of a role in your classic type of chemistry, they play extremely important roles in the chemistry of life. And that's probably a good place to take a temporary break. And so I'd like to say 10 minutes, but